On the paper, bacterial membrane vesicles transport their DNA cargo into host cells. This is the abstract of the paper. The main focus of the paper is on bacterial outer membrane vesicles and their contents. Bacterial outer membrane vesicles, or OMV, contain biologically active compounds, including DNA. However, not a lot is understood about this DNA and whether or not it can be transported between bacteria or into host cells.
For this experiment, five different gram-negative bacteria were utilized from 3 different classes listed and shown here. These are all common disease-causing bacteria from which antibiotic resistance and virulence factors can occur. What specifically was studied from these bacterial types were the outer membrane vesicles from exponential phase cultured cells. They were isolated and studied to test if the OMV-associated DNA presence and location was the same across all gram-negative bacteria. After this was completed, they then focused solely on Pseudomonas aeruginosa, which is why I have it underlined here. And this was specifically studied in regards to, again, the DNA presence and location, just to confirm the previous studies. And then they continued to also study when these OMVs are released and their characterizations and also their transport abilities.
Small membrane vesicles have been conserved in both multi- and unicellular organisms throughout evolution in their roles to facilitate intercellular communication and help transport molecules. These membrane vesicles can typically be found the same components as its parent cell. In gram-negative bacteria in particular, it was found that the outer membrane vesicles were enriched in specific membrane components and virulence factors that it's believed outer membrane vesicles may be a form of selective packaging. These outer membrane vesicles have been found to be produced both in vivo and in vitro, but especially during times of stress. Lice bacteria release membrane fragments that go on to form membrane vesicles and as a result engulf cytosolic contents such as DNA. While OMV-associated DNA and host pathogen interactions are poorly defined, it can be seen how they help with the exchange of inter- and intraspecies DNA, just like with the lice membrane vesicles. This transfer of DNA contributes to transfer of antibiotic resistance genes within bacteria. OMV-associated DNA can aid in host colonization of bacteria because it has been found they play a role in biofilm formation as well.
Originally,
the goals behind this paper are as follows: to learn more about prokaryotic membrane vesicles in general, to understand the characteristics of DNA found in outer membrane vesicles, as an increased number of studies that prove DNA in membrane vesicles do not provide enough information on the matter, to better understand the interactions and roles of outer membrane vesicle-associated DNA and hosted pathogen interaction. And the final and overall goal of the paper is to confirm if it's through outer membrane vesicles that DNA can be transported from the bacteria to the host cells, how it can be tested, and to find the mechanism through which genetic material can be exchanged between prokaryotic and eukaryotic organisms.
Originally, our presentation was supposed to fall along the timeline with fractionation as the technique of the week. In this paper, they utilized a sucrose gradient fractionation, and this is on the subcellular level, so it is going to be a little bit different. The cultivation of the bacterial strains and the isolation and purification of the OMVs came prior to this specific fractionation described. This can be seen in the diagram to the right where the cells were cultured in the first step. And in the case with the paper, it was specifically cultured in mid-exponential phase. After the culturing, they were then filtered and isolated by ultracentrifuge, which is seen as steps 2 and 3 on the right in the figure or in the diagram. They then were resuspended in PBS, which is a phosphate buffer saline, before the sucrose gradient fractionation was begun. The fractionation was of P. aeruginosa. Once the culturing, isolation, and purification of the OMVs was done, the density gradient of the OMVs could be completed. The OMVs were washed 3 times in PBS and then layered onto a discontinuous sucrose gradient, which separates the OMVs based on the density with different percent sucrose and weight by volume. A discontinuous gradient just means that all the separated components are distinctly fractioned, as seen in the diagram in the bottom left corner. The separation was done by ultracentrifugation, and then the proteins and DNA concentrations of each fraction was measured. The proteins were assessed by a Western blot, and the DNA was visualized on a 0.8% agarose gel. This fractionation was done to see where OMV-associated DNA was located in relation to all OMVs. In this case, with the bacteria strain done specifically, the DNA was mainly found in smaller sized OMVs, which correlated with fractions 4 through 6 of the completed sucrose gradient fractionation. Here is the first figure in the paper. This portion of the research utilized all 5 different gram-negative bacterial OMVs mentioned, which is also listed to the left of the images and diagram and can be compared in each row. So in part A of this figure, it is showing the isolated OMVs from exponential phase cultures, which is shown by transmission electron microscopy and is the first column of images seen in this figure. And what exponential phase means is that the bacteria is just regularly dividing by binary fission. And also the arrows in part A indicate flagella, which is seen in 2 of the images, so in 2 of the bacterial images. And in B, there are 2 columns for this portion, which are both stained to show DNA. The first column of B is without the DNase treatment, and the second column of B is with the DNase treatment. The DNase treatment is used to remove the external DNA, leaving only the internal DNA left over. So the first column of B would include both internal and external DNA, whereas the second column of B would just be the internal. Then finally, the very last column of this figure, which is C, is just the quantification of the internal and external OMV associated DNA, which is shown in bar graphs. And this is basically just used to sum up the results that we can see in the images and divide up the exact concentrations of where the OMV associated DNA is located within the OMVs themselves.
The figure shows super-resolution microscopy of bacteria gathered during the mid-exponential phase of bacteria growth, a 4-hour incubation period. The figure on the left was tagged with a lipophilic stain that targeted for outer membrane vesicles, resulting in the green color before you. The middle figure was an adapted click EDU assay that labels nascent DNA within dividing bacteria and produces a red color before you. When the 2 figures are overlapped, it can be seen that they are nearly identical. The outer membrane vesicle figure being bound to a lipophilic stain in the same area where bacterial DNA is dividing confirms that bacterial DNA is associated with outer membrane vesicles released by dividing bacteria.
The purpose of Figure 5 was to determine if the OMVs and the bacterial DNA contained within them are capable of transporting themselves inside of eukaryotic cells. The top row of Figure 5, Figure 5A, shows a series of different images. The leftmost image is the nuclei of lung epithelial cells, stained blue with DAPI. The image directly to the right of that shows the OMV stained green with DO. The next image shows the bacterial DNA stained red with EDU. The final image is a merged compilation of the other three images, which, as you can see, shows co-localization of the OMV in the eukaryotic cell. Figure B shows the same thing as Figure A. The only difference is the OMVs were first treated with the DNAs to remove any exterior bacterial DNA. Figure C through F are varying different controls relating to the bacterial DNA marker EDU. EDU is a two-part cellular marker. You have the EDU marker, and then you have two other components that allow for appropriate visualization of the marker. Figure C has no EDU added, but the two markers that allow for visualization have been added. Figure D has only one of the two markers added and EDU added. Figure E has no EDU added at all. And Figure F has EDU added, but the membranes are non-permeable. The final conclusions of the paper were: OMVs are used by bacteria for bacterial intercommunication and also for sharing helpful genes like antibiotic resistance. The OMVs and the bacterial content most likely play a role in pathogen and host interaction. The OMV DNA may modulate host cell responses in order to avoid detection and destruction by the immune system. And the findings may have implications for using the OMVs to induce an immune response beforehand, as with a vaccine. And these are our references.
Here I have provided a link to the worksheet that goes along with the presentation, and below that I also have a link to the key to this worksheet.